Hello everyone. Today we will be discussing on pathogenicity and clinical manifestations by Staphylococcus as a genus. Staphylococcus genus contains many organisms like Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus. So these are all the common cell of skin and nasal membranes, and common cell is the one which is not causing any discomfort or problem to the host. But these organisms can become opportunistic when there is some disturbance in the host, maybe due to some disease or some kind of wound infection, or it's an immunocompromised host. So, under these conditions, this pathogen can become opportunistic, and it causes lot of diseases and infections, and which can be clinically seen. So we will study its clinical manifestations also. So basically, the pathogenicity is due to various virulence factors, and these virulence factors are the factors which invade host immunity. So if you want to know what is immunity, you can go to my previous videos where I've put lot many videos on the immune system itself. And if you want to know more about virulence factors, that too, I have put a video on virulence factors of Staphylococcus aureus, basically. So you can go and see that too. This Staphylococcus genus is important genus causing lot many of infections because it is also capable of forming biofilms. What are biofilms? We will be studying later in this video itself. And this group has multi-drug resistant also. So it has emerged as multi-drug resistant organism. So what is multi-drug resistance? If you want to know that, I have put a video on that too. So you can subscribe my channel and see all the videos on it if you are interested in small things which I am talking about. I have put short videos on the things. So you can go and watch that. So what coming back to Staphylococcus genus, which has many organisms which causes lot many infections. What are the factors which help them in invading immunity of host? It can invade both kind of immunity of host, that is natural and acquired one. So, first of all, it has a polysaccharide capsule. So, this polysaccharide capsule protects it or mask this from phagocytosis. So, this bacteria, when comes in contact with the cells of immune system, what are the different cells of immune system? You can go back to my video and watch it on my channel. So, here I'm just calling them cells of immune system which perform phagocytosis. So, this bacteria can fool those cells because it is wearing a mask, and this mask is covering all the foreign things which can be detected by phagocytes for phagocytosis. So this bacteria is wearing a mask and entering inside the body and fooling the immune system. And another thing which this bacteria have is extracellular fibrinogen binding protein, and this also has a potential to block phagocytotic uptake. So again, this fibrinogen binding protein, which is present, extracellular fibrinogen binding protein, which is present on the bacterial surface, is avoiding phagocytosis uptake. Then there is one more protein A present, and this protein A fools the acquired immunity by capturing immunoglobulin G in inverted position and thus suppresses the B cells response. So what are B cells? What is acquired immunity? What is immunoglobulin G? You want to know all of this, you can go and subscribe my channel and find it in different videos. So here what I want do not break the continuity of the topic. What we are discussing is that Staphylococcus as a group have certain number of microorganisms, certain number of bacteria which can cause infections in humans. And it can cause infection in human by fooling the protective system, yeah, defense system of human body. So defense system of human body is maintained by two types of immunity, that is a natural and acquired. So this bacteria is capable of fooling natural as well as acquired immunity, because it is having a polysaccharide capsule, extra fibrinogen binding, extracellular fibrinogen binding protein, and protein A both. So first two is fooling phagocytotic cells, and protein A is fooling B cell response. That means B cell response is acquired immunity. And other factors are like exoenzymes. Various enzymes are present in this bacteria, that is catalase, coagulase, fibrinolysin. Catalase is not present in this bacteria. Please, catalase, this bacteria is catalase negative. So catalase is present in strepto. Sorry for this. This is not present in this bacteria. Catalase is present in streptococcus. Please, you just delete this catalase. Coagulase is present, fibrinolysin, hydrolinidase, beta-lactamase, DNase. These are certain enzymes which are present in this bacteria. They cleave in and inactivate various immune defense and surveillance molecules. Our immune system is there to protect us, and a continuous surveillance is going on to detect the foreign entries inside the or invading inside our body. So this bacteria is able to fool because it has, please delete this catalase, it do not have catalase, but it has coagulase and other enzymes mentioned there. So by these enzymes, it is fooling our complement factors, antimicrobials, and surface receptors that are important for leukocyte chemotaxis. Chemotaxis is movement of leukocyte towards the foreign things. But these enzymes present in this bacteria are stopping the fooling our immune system, and they can even disrupt or cause damage to endothelial and epithelial barriers. So epithelial cells are also first line of defense, and endothelial cells, it can also damage those cells and proliferate through and enter through them. And this bacteria have toxins also. These toxins are also the provide is one of the factor which provide entry inside the host, or this is factor which is invading the host immunity. So we have all these like polysaccharide capsule, fibrinogen binding protein, protein A, exoenzymes, and toxins are the different factors helping in invading the immunity of host. Now, coming to toxins. Toxins are the factors which are invading the immunity of host and masking the bacteria and letting the bacteria enter inside the host and cause damage. So these toxins are of three classified under three major groups. One is the pore-forming toxins. That means it forms pores in the cells of the host, immune cells of the host, as well as RBCs, red blood corpuscles. So Hemolysins, that is alpha hemolysin, that is also called alpha toxin, hemolysin B, leucotoxins. Under this, there are 4 different types of leucotoxins. One I have mentioned here. Then phenol soluble modulins are also the ones which are forming pores inside the host cell. Then exfoliative toxins have another role. We will discuss it. And super antigens are also present. I have put a full lecture on super antigens. So if anyone who's interested in knowing what are super antigens, please you can go to my other video and see. Now coming to back to hemolysins alpha, they affect, they are present, and they are, as the name suggests, hemolysins, they must be lysing RBC as well as they lyse epithelial cells, endothelial cells, T cells, monocytes, and macrophages. So they are affecting these cells, and they are damaging, producing pore in these cells. So as they can damage endothelial cells, epithelial cells, they can cause severe tissue damage, and they are invasive in nature. Then they can invade the go to different tissues. Next is hemolysin beta, or you call it sphingomyelinase also. That is also cytotoxic towards human keratinocytes, polymorphonucleoleucocytes, monocytes, and T lymphocytes. So it is damaging all these cells. and it inhibits interleukin 8 also and expression of interleukin 8 leads to damage the endothelial cells too, and it's said to contribute towards phagosomal escape and induction of biofilm formation. So the strains which are having hemolysin B, molecular biology has made it possible for us to see which strains are expressing which molecules. So this hemolysin beta, or you can call it sphingomyelin C, also sets to contribute towards biofilm formation. Remember that. Then leucotoxins are of 4 types, that is gamma hemolysin, leuc-ED, leuc-AB, or GH, and Pentane-Valentine leucosidin. PVL is the most commonly known toxin. All of these toxins lies or create pores in the leukocytes and in the cells of leukocytic lineage. They kill neutrophils also, while gamma hemolysin and leuc-ED have lytic activity against RBC also. So this was about leukocytes, and in this, one of the leukocyte is Pentane-Valentine leucosidin, and it affects, as the name indicates, it's leucosidin. That means it kills leukocytes and cause tissue necrosis. It is associated with formation of furuncles, cutaneous abscess, severe necrotic skin infections, and Pentane-Valentine leucosidin genes not detected in strains responsible for that. You have to remember endocarditis, mediastinitis, hospital-acquired pneumonia, UTI infection, urinary tract infections, enterocolitis, and toxic shock syndrome. All this, in this case, PVL is not detected. These for the people who are going for entrance exams where multiple choice questions are there. So remember that PVL genes are at present not detected in the strains which are causing these diseases which I have mentioned below. So phenol soluble modulins are another pore forming toxins, and they are said to be detected in the strains which have invasive character, which can invade tissues and which can lyse human neutrophils and stimulate inflammatory response. So phenol soluble modulins are this. Then exfoliative toxins, ETs, also known as epidermolytic toxins, and are exotoxins. Remember that Staphylococcus secretes both kind of toxins, exotoxins as well as enterotoxins. So ETS is expressed in Staphylococcus aureus, and it is related to localized epidermal infections such as bullous impetigo, Staphylococcus scalded skin syndrome. We will talk about this later. What is this? But remember, ETS is responsible for this. Then superantigens. Superantigens, what are superantigens? Please go to my previous videos and see. While 23 Staphylococcus superantigens have been reported, one of them example is TSST1, staphylococcal enterotoxins, superantigen-like toxins, and diseases such as sepsis, skin and airway allergies are caused because of superantigens present in Staphylococcus. What are the different clinical manifestations of this group or genus? Genus, Staphylococcus genus, most strains of Staphylococcus aureus are reported to express fibrinogen or fibrin binding protein, which promotes attachment to blood clots and traumatized tissues. So whenever there is some tissue injury or there is injury, epidermal injury, then this, through these injuries, since this Staphylococcus bacteria are common cell of the body, they can enter and they can reproduce through these. And then bacteria gain access to host through damage to that I have already told, skin or mucosal or tissue sites, and colonize by adhering to cells or extracellular matrix, evade host defense mechanism, multiply and cause tissue damage. So that is how they enter inside. I have told you how they, I have put explain in initially also what mechanisms do they have to evade immune system of the host. And then once they enter inside the, if we avoid phagocytosis and all, then they start releasing exo and enterotoxins. So what kind of infections they can cause? They can cause folliculitis. I will show you the picture of folliculitis. It's pimple-like, but I will show there is a difference between uh, simple pimple and folliculitis, but I'm going to show it's pimple-like structure. Then if there are boils, it is furuncles. Bunch of boils together is carbuncles. Diffuse skin infection, that is superficial impetigo, and infection of dermis, cellulitis, that also I'm going to show you the pictures, because just naming them will not help. And deep infection, subcutaneous abscesses also cause. So these are different kinds of infections which can be caused by staphylococcal strains. So localized infections, diffuse skin infection, and deeper infection. So coming to folliculitis, this is usually seen, this is the pimple like structure seen near the follicle of hair follicle. Then furuncles are boils, which is seen like a boil on the skin. And superficial impetigo, when all these together like this, boils are together, they causes and superficial impetigo. So you can see these in on the chin together. Then going to the next one, cellulitis, which is caused by Staphylococcus. It is here you can see a swelling. This structure will be warm also, and this kind of inflammatory response, inflammation is also seen. This is cellulitis. When we talk about Staphylococcus scalded skin syndrome, here you can see skin is removed, peeled off. This is scalded skin syndrome. Fluids comes out and skin is peeled off. Toxic shock syndrome is this radish appearance. It has with these. This is I'm showing on the skin the appearance, radish appearance, and skin removal is removed, and this. Watery fluids are coming out from the tissues. These are seen on the skin, and they are multi-system in is also involved in this. That means there will be diarrhea, vomiting, fever. This will occur with toxic shock syndrome and scalded also. With cellulitis also, fever will be there, chills will be there. So other symptoms are there, but on the skin, all these things will be seen. This is the characteristic of Staphylococcus group. Coming to subcutaneous abscess, which I have told that Staphylococcus also causes subcutaneous abscess, that is collection of pus that's walled off and sometimes develop thin walls within it. So there will be a collection of pus, and then that skin will be peeled off, and you can see partitions of skin filled with pus. So this can happen all over the body, and if it's happening on the muscle, then it is called pyomyositis. If in the bone, then osteomyelitis, joint septic arthritis, blood septic thrombophlebitis. So you can see Staphylococcus can cause all these kind of infections also. But characteristic feature will be subcutaneous abscess in these cases. This bacteria can also enter in the blood, as it has invasive character. Also, it enters through in the blood and reaches to different organs. So when it's causes sepsis, that means blood vessels expands, blood pressure falls, poor perfusion to various organs happens. It can go to brain, causes bacterial meningitis, lungs, pneumonia, epidural abscess, endocarditis, myocarditis. Reaches if it reaches to heart. So it can cause multi organ failure also. It can go to different organs also. Let's come to all the toxin mediated diseases, that is scalded skin syndrome, Ritter's disease, toxic shock syndrome, and food poisoning. Characteristic feature of food poisoning of Staphylococcus, it can appear within 8 to 30 minutes, 8 minutes to 30 minutes of having food if staphylococcal toxin is there, and this is self-limiting. Within 1 day, a person will improve. So then this bacteria also have a character to form biofilm. How the biofilm are formed? Because it's a common cell, it is present on the skin, and when certain prosthetics or valve or catheter is introduced inside the body, this bacteria reaches to and start growing near these things which have been inserted, and once it gets inside that, then it starts forming extracellular matrix, which is made up of exopolysaccharides. And I've already told you that its polysaccharides are one of the things which can fool the phagocytic cells, and then these bacteria communicate with each other through biochemical signals, and they start maturing there, reproducing, and maturation occurs. So biofilm formation has two major steps. One is attachment and other is maturation. And once these bacteria group get through biochemical signals special genes, and the specific character of staphylococcus is that it do not allow other bacteria to grow with it within its biofilm. While pseudomonas has they can grow with other bacteria, but staphylococcus avoids all other kind of bacteria, and its own colonies grows. And once they mature, they start breaking up and dispersing and taking different routes and colonizing there. So this biofilm is also very dangerous. While this bacteria is this staphylococcus group is multi-drug resistant too. So why it is resistant? If you want to know that, you can always go back to my different videos which I have put forward. Hope you would have understood this properly. Thank you so much.